biotechnology refers to biological systems and processes to develop products that improve human life and quality of living.
Transgenic organisms are organisms whose genome is composed of DNA from at least 2 distinct species.
Peekaboo. They are commonly used to provide solutions for malnutrition and agricultural waste. However, transgenic organisms are also used to model human immune responses and diseases. You may be wondering, how are they made? How are transgenic organisms made? That's a great question. Let's find out. First, DNA sequencing is used to determine the order of nucleotides, locating both start and stop codons. These genes are then tested for their functions by modeling the effects of removing these genes. But what next? Scientists need to isolate the genes. To isolate the gene, the DNA must be separated from other components of the cell. This is done by a process called centrifugation, a high-speed spinning process where substances are separated based on their physical properties. Restriction enzymes are then used to cut the desired gene from the DNA sequence. Fragments are then sorted by size using gel electrophoresis, where a DNA probe is used to identify the desired gene. They are then cut out of the gel and amplified using PCR, polymerase chain reaction. Okay. Polymerase chain reaction amplifies the gene by putting it through thermal cycles. First, DNA is heated to 95 degrees Celsius to ensure the breakage of the hydrogen bonds between complementary nucleotides. Then, the DNA is cooled to 55 to 72 degrees Celsius, facilitating the binding of primers on the single strands of DNA. DNA is then heated to 75 to 80 degrees Celsius, optimizing the activity of Taq polymerase. This, therefore, promotes the DNA replication / synthesis. This cycle, or these cycles rather, are then repeated until there is a sufficient amount of copies of this gene. The restriction enzymes cut DNA at specific locations, leaving sticky ends. These sticky ends anneal with bacterial plasmid upon their assembly. The gene and plasmid bond via hydrogen bonds. The bacterial plasmid is then cut with the same restriction enzyme used to cut the desired gene. Therefore, sticky ends of the plasmid and the gene are complementary. This cutting of the plasmid opens the plasmid ring, creating a space for the desired gene to get inserted. DNA ligase then catalyzes the formation of phosphodiester bonds between the plasmid and the desired gene. The modified plasmid is then inserted back into a bacterial cell, where it replicates until a sufficient amount is produced. To transfer this transgenic bacteria back into the host organism, there are 4 main methods. First, microinjection. Second, biolistics. Third, electroporation. And fourth, transduction. After fertilization, the organism possesses the selected gene in their genome, altering their phenotypic expression. Therefore, the process is complete. So how did we get to a point where we could create these organisms? Good question. Several major discoveries and innovations have led to advances in technology that have allowed us to create transgenic organisms. First came the discovery of DNA structure in 1953, a discovery made by Rosalind Franklin and Maurice Wilkins, who questionably shared their crystallography images to John Watson and Francis Crick. Then, in 1962, Osamu Shimomura isolated the green fluorescent protein, creating an indicator for DNA. DNA ligation was discovered in 1967 with research from Gellert, Lemon, Richardson and Hurwitz laboratories, where DNA ligase catalyzing the formation of phosphodiester bonds was discovered. Then, in 1968, Werner Aber noticed that bacteriophage infection was fought off by bacterial strands chopping up the DNA. The first isolated restriction enzyme was EcoK1, which was isolated by Matt Messelson from the bacteria E. coli. In 1970, Hamilton Smith was working on HIND-2 enzymes, assisting his understanding of restriction enzymes. By 1972, he had purified these enzymes, offering a way to cleave DNA at specific loci. Then, in 1971, Paul Berg used EcoRye to cut DNA from the virus SV40 and lambda phage. The restriction enzymes left sticky ends which were complementary and annealed upon assembly. Hence, the first recombinant DNA was formed. Daniel Nathans and Kathleen Danner used HIND-2 enzymes to cleave the SV40 DNA into 11 fragments, which were separated by polyacrylamide gel electrophoresis. This allowed the linear order and relative positions of cleavage sites to be determined, allowing the mapping of DNA. In 1981, Thomas Wagner led a team who inserted the rabbit beta-globin gene into a mouse, which passed on the gene to its offspring, making it the first successful transgenic organism.